Hi, welcome back to Educator.com. This is Laboratory Investigation 8, Genetic Transformation. Alright, now for an introduction. The purpose is to genetically transform E. coli, it's a bacteria, so that it glows green when exposed to UV light. That's pretty awesome. Now this is taking advantage of another organism, a kind of sea jelly or jellyfish, that has that protein that allows it to glow naturally, and when this is moving around in the ocean and absorbs UV radiation from the sun, the way it interacts with the protein gives them this greenish glow, and it serves them a purpose in their environment. Now the bacterium does not naturally have it, but when you take that DNA out from the sea jelly's genome and put it in a little plasmid, a little circular piece of DNA, and introduce it to the bacterium, you can get it to accept that DNA and express it. And that's the key to genetic transformation, literally transforming the genetics of an organism. So materials, 5 agar plates, this would be per group, but for one group, 5 agar plates. Now agar is a nutrient source. It's actually from a kind of seaweed, and it kind of looks like plain jello with a yellowish tint. You put it in petri dishes, and you would usually take the powdered version, dissolve it in liquid, heat it up, pour it, let it cool until it solidifies. You would need an E. coli starter culture. Of course, you would need to do this under the supervision of someone with experience, a teacher or professor, because typically with this lab, you would order E. coli that is, not as harmful. There are strains of E. coli that typically don't kill a person or make a person sick, and that's what you use for this lab. Now, you still don't want to get E. coli, even from the harmless ones, in your mouth, so you have to be careful. So a starter culture to get the colonies going, ampicillin, which is an antibiotic, arabinose, a kind of sugar that helps this lab work, plasmid. Now, this is not something that's easily found. You would have to order this from a company that provides the plasmid, and the plasmid has AMP resistance. What I mean by AMP resistance is resistance against ampicillin, so it allows the bacterium, when exposed to the plasmid, to not die from this antibiotic, and it allows them to make what's called the GFP, which I'll mention again, the green fluorescent protein from the gene that was originally in the sea jelly. Inoculating loops, they look like, they're either metal or plastic. They look like long sticks that have looks like a little ring at the end, and they're really good for it's like making a starter culture where you take a little bit of bacteria from kind of an aqueous area, like in a vial, and then you streak it on the plate. And if you were to do this lab in real life, you would follow those directions to maximize the formation of those colonies of bacteria. Pipettes to suck up the materials from the vials you would be using to get the ampicillin, arabinose, etc. Medicine droppers is the same thing. An incubator, heating up these plates to a certain temperature will maximize the cell division of the bacteria and make this lab come to fruition a little bit faster, and a UV light for visualizing that green glow. Time required about 5 days from the point where you have your starting culture of E. coli to the point where you actually can see the glowing. Sometimes you can have it happen in as little as 3, but it really depends. And like I kind of hinted at earlier, with having a teacher or professor supervise you, there's some risk of getting ill or worse with this lab. So gloves and goggles would be required, so that if you do get something on your hands, it's not going to harm you. Before you put your hands in your mouth, dispose of the gloves, wash your hands thoroughly, use hand sanitizer, and make sure that nothing terrible happens during this lab.